thank you for joining me. I'm Dr. Hensley, and today we're going to be talking about the central dogma of biology. The central dogma has two main parts, transcription and translation. The central dogma describes how hard copies of genes in DNA are eventually made into functional proteins so that the cell may grow and divide. The central dogma has two main parts, transcription, where DNA is taken to RNA, and translation, where RNA is taken to protein. In transcription, DNA is taken to RNA. These are both nucleotides. They look very similar chemically, except for they have a few minor differences. These differences mean that DNA likes to pair up with itself and make this double helix structure you may have seen in biology class, whereas RNA likes to be a single strand by itself. The second major difference is that one of the nucleotides is T in DNA, but the same nucleotide in RNA is known as uracil or U. Because DNA is a more stable molecule, it contains all of the genetic information in the cell. Think of DNA as the internet, whereas RNA is the research paper you would write from your sources on the internet. You don't need all of the sources of the internet to make your research paper, just a few. Transcription starts when little proteins called cofactors are signaled to bind the start of a gene. They act as signal flares to bring in a much larger protein complex called RNA polymerase. It is RNA polymerase's job to actually copy the DNA gene. It does so by moving along the gene itself. As it moves, it produces a single strand of messenger RNA, which matches the strand of DNA. Once RNA polymerase has done its job, it falls off the strand of DNA, and you are left with your original DNA genetic copy and your new RNA copy. In translation, you're taking the nucleotides of RNA and making it into a new form of information called amino acids and protein. This is a lot like if we were taking our English essay and making it into a Spanish essay. Translation begins when the messenger RNA attaches to the ribosome in the cell. The ribosome recognizes a series of 3 nucleotides called a codon. The ribosome brings in tRNAs, which recognize each codon and at their end have an amino acid. As the ribosome moves down the messenger RNA, new codons are recognized and new amino acids are added to the end of a growing peptide chain. As the ribosome moves along the messenger RNA, the protein chain grows, the tRNAs continue to fall off, and eventually, once you've reached the end of the messenger RNA, you have your complete protein. So to review, DNA is transcribed into RNA, RNA is translated into protein, and this shows us how all of our genes are made into protein so our cells may function. I know we went through the central dogma really fast, so if you have any questions, please check out the links below or leave me a comment and I'll get back to you in a timely manner.